The overall goal of this methodology is to rapidly quantify abdominal pigmentation in the fruit fly Drosophila melanogaster.
This method can help answer key questions in the field of evolutionary biology, such as identifying the genetic loci that underlie, in turn, intraspecific variation in morphology, and understanding the development and evolution of morphological phenotypes. The main advantage of this technique is that it allows users to non-destructively measure various aspects of abdominal pigmentation, including the spread and depth of pigmentation.
Though this method can provide insight into Drosophila melanogaster's abdominal pigmentation, it can also be applied to other Drosophila species and easily adapted to measure pigmentation patterns in other insects and animal taxa.
Prior to imaging, the flies can be stored in 70% ethanol to maintain their pigmentation. Visualization requires a mounting pad. Load a 60 mm Petri dish with about 10 ml of 1.25% agar in water. Under a microscope, use fine forceps to dig a 20 mm long, 2 mm wide, 1 mm deep trench into the gel. Several trenches can be made in one dish. Then, embed flies into the trench, with the surface for analysis sitting just above the gel surface. In this case, the dorsal side is of interest. Now, fill the dish with 70% ethanol until the flies are completely submerged. This reduces reflections off the cuticle that can obscure the pigmentation pattern. Now, start the imaging software and digital camera connected to the microscope. For a light source, cold light from a double gooseneck device is ample. In the image analysis software,